Hello everyone. In the last video, we have started with the biomolecule chapter, wherein we have read the introduction part of the molecules and the carbohydrates. Now, in this video, we are going to start with proteins. So first, coming to the meaning of the proteins. Proteins are called heteropolymers. Now, what do you mean by heteropolymers here? Hetero means different. So that means they have got different monomers. Now, if you talk about the monomers of the proteins, what are the monomers of the proteins we have? Let me write down here. Monomers of the proteins are nothing but amino acids. So here you have to remember that whenever the amino acids are forming the proteins, say these are the amino acids which are involved in protein formation. So all the amino acids will be different here. Let us say first one is methionine, second one can be cysteine, third one can be phenylalanine, fourth one can be tyrosine. So like this, we have different types of amino acids which will form our protein molecule. And hence, the proteins are called as heteropolymers here. Now, in our body, there are 20 amino acids which make whatever proteins we have in the body. Yeah. Now, coming to the bonds which are present between the amino acids, these are called peptide bonds. So, in this topic, we are going to study the different types of amino acids. What is the structure of amino acid and what is the bond? I mean, how is this peptide bond found between the or formed between the amino acids? So all these things we are going to cover in this topic. So first, coming to the amino acids, we know amino acids are the monomers of the proteins. Now, these amino acids are called substituted methanes. Now, what do you mean by substituted methanes? If you consider the structure of methane, it is CH4, isn't it? This we already know, CH4. So let me elaborate the CH4. Say now, this is my CH4 again, isn't it? Now, I said it is substituted. That means the hydrogens of the methane will be substituted with some other functional group. That is the reason why it is called substituted methane. So if I explain this here, now here we have hydrogen. So let me consider this hydrogen. So here, this hydrogen is replaced by one carboxylic group. So here it is carboxylic group. Now this hydrogen, I'll be replacing it with one amine group. So this one is my amine group. Okay, now this hydrogen, I'll keep it as it is. Whereas the fourth hydrogen, I will replace with a variable group, which I call it as R. So R here will be my variable group. Now, what do you mean by variable group? This group or this functional group will be varying from one amino acid to another amino acid. So if you consider any structure, any amino acid structure, all the three, that means amine group, carboxylic group, and this hydrogen, these three will be same for all the amino acids, whereas the different 20 amino acids what we have discussed is different only in this R group. For example, now here you have to remember the amino acid structure, because in the exam they can confuse you this structure with some other, like for example, we have one carbon to which hydrogen, amine, and carbon, COOH, here R group, here hydrogen, and here hydrogen. This is given in the one of the options in the exam. Now this is not an amino acid. All right. Why it is not an amino acid? Because you have to remember that amino acid is a substituted methane. So they can confuse you this structure with this structure. Clear? What is the difference here? In amino acid, a single carbon will be bonded with all the four different functional groups, isn't it? One carboxylic, amine, then hydrogen, and an R group. Whereas in this structure, what did I draw? There are two carbons. Now, first carbon is bonded with amine group here, whereas second carbon is bonded with carboxylic group. All right. So this is not an amino acid. Very important. It is not an amino acid. Please remember. An amino acid is a structure wherein one carbon will be attached to both carboxylic as well as amine group and the hydrogen. Only the fourth hydrogen will be replaced by the R group. So this structure will be called as amine group. Clear? You should not get confused between this and this in the exam. Now, coming to the examples. So if I give you the examples wherein we have different types of amino acids, I said each amino acid is different only with respect to R group. So let me consider a few examples here. Let me take the simplest amino acid. Say this is one carbon to which carboxylic acid is attached, amine as attached, hydrogen, and the R group is only hydrogen. So if this is the case, now here the example is nothing but glycine. So glycine is one of the amino acid, and we can say this is the most simplest amino acid we can have because the R is simply hydrogen. Clear? Now, if I replace the R group with some other molecule, so let me write down that. Now here, if I'm writing methyl group, so if R is replaced by methyl, now this is our alanine. Clear? And one more example, if I give you, let me write here. You can draw without seeing if you want to practice the structure of amino acid. That will be very helpful for you. So here, coming to the R group. So if I replace it with alcoholic functional group, now here this will be your serine. So serine is an amino acid which have alcohol as its functional group. So these are the three examples which are also mentioned in your textbook. So you have to learn these examples. Clear? Structures you have to learn. If you look at the structures given in your textbook, now these are the structures given in your textbook. Glycine, alanine, serine. Clear? If you observe the structures here, to one carbon, all the three functional groups are attached. That is COH, COH, COH. This is common. NH2, NH2, NH2. This is common. Hydrogen, hydrogen, and hydrogen. All these three are common for all the amino acids. Clear? What is difference here? The R group. For glycine, it is H. For alanine, it is methyl group. For serine, it is CH2OH. That means a hydroxyl group is attached. Clear? So this is about the different examples of amino acid. We have so many different examples. We'll talk about that later. All right. Now, I just wanted to show you the structure of amino acid by using these examples. Now, coming to the bonds which are present between these two amino acids. So let us consider the peptide bond formation. Now, how is this peptide bond formation? For that, again, let me draw the amino acids first. You can draw the amino acid without seeing. First, we'll draw the structure of glycine, and then I'll draw the structure of, let us take alanine. Okay. So glycine, again, COH. Now here we have NH2, H, and here H. Clear? If I talk about alanine, again, COH, NH2, H, and CH3. Clear? So let me elaborate this so that it will be helpful for you to understand. So NH2, I can write like this also. Say here N, H, and one more H here. Okay, I can write like this, isn't it? So this is glycine and the other one is alanine. Now what I have to do for peptide bond formation, I have to remove one water molecule between these two. Clear? So let me write down that first. One water molecule. If you consider the water molecule, we have two hydrogen and one oxygen. That means I have to remove two hydrogens between them and one oxygen. So from where exactly I'm going to remove? OH from glycine and one of the H from amine group. Clear? OH of glycine and one of the H from alanine, I will remove in the form of water molecule. Clear? And now I can show the peptide bond formation. So what is left here? Let me write down that first and then I'll tell you. Say this is COO. So COO I can write like this also. C double bond O. Then here. Okay, so I'm left with this structure. And here if I talk about alanine, NH, isn't it? One H is removed. So I'm left with one H. Then again H, COOH, and here methyl. So between them, I'm getting the peptide bond. So this is how we get peptide bond, by removal of one water molecule between the two amino acids. Clear? Now you can try with one more amino acid. Let us try with one more amino acid. We know the structure of serine, isn't it? So let me draw the structure of serine and then we'll remove the water molecule. Please draw without seeing so that you can easily understand. Clear? So let me draw the structure of serine here. It will be a practice if you draw without seeing. And NH, here also H, CH2OH. So this is the structure of serine we have learned. Clear? Now I have to remove one water molecule between them. So OH of alanine and 1H of serine I will remove in the form of water. Clear? Now what is the bond I'm getting here? The bond is called peptide bond. So let me draw the remnants, what I get from this. So NH, C, COOH here, then hydrogen, CH2OH. This is what I'm left with. And this side, I have removed OH. So let me cut out this OH. Now I can form the bond between them like this. Clear? So this is my peptide bond formed between what? Alanine and serine. Clear? So this is how we get the peptide bond formation by removal of one water molecule between the amino acids. Now, if you observe the chain of amino acids, let us say this is an example of a protein which is formed of 3 different amino acids. Clear? So in this first amino acid, which is glycine, here the amine group is free, isn't it? Here the amine group is free. And if you consider the last amino acid of the protein, now in this example, the last one is our serine. This is our serine. So which functional group is free here? This one is free. And this is what? Our carboxylic group. Clear? So whatever protein you take, clear, whatever protein, how much ever the number of amino acids are there. Let us say it has got hundreds of amino acids or thousands of amino acids. So whatever protein if you take, the first amino acid, if you consider, always the amine group will be free. Whereas the last amino acid, if you consider, there the carboxylic group will be free. So with this, what we can do? We can determine the ends of the protein. Isn't it? We can say which is your first amino acid and which is the last amino acid by looking at this. So here, the left hand side. Now this is my left hand side, isn't it? This is my left hand side. And this is my right hand side. So always the left hand side will have the first amino acid and the right hand side will have the second the last amino acid. Left hand side, here we have the first amino acid, which we call it as the N-terminal. This point you have to remember. Why it is N-terminal? Because this N I have taken from the N of amine group. And the last amino acid, we call it as C-terminal. Clear? So this is very important, like which end represents C-terminal and which end of the protein represents N-terminal. N-terminal, if you talk, that will be forming your first amino acid of that peptide chain. If you consider C-terminal, so this will be your last amino acid. These points you have to remember. N-terminal always the first amino acid, whereas C-terminal will be the last amino acid. Clear? I hope this is clear for you, like why we call it as N-terminal. So please remember N we have taken from the amine group, and here C for the last amino acid we have taken from the carboxylic group. So this is about the terminal ends. The first amino acid is called N-terminal and the last amino acid we call it as the C-terminal in any peptide, polypeptide. That means any protein. Clear? So this is all about the formation of your peptide bond and the terminals of a protein. Now we'll talk about the classification of amino acids. That means what are the different types of amino acids. So the first type of classification is based on whether the functional group, that is the variable group, has got acid in it or base in it or neutral. Clear? For example, if you consider the structure of, say, glycine, or we can have alanine, or we can have serine. See, all this we have just read now, isn't it? So this will be very easy for us to understand. So glycine, alanine, serine structure, if you see, what are the structures here? We have carbon to which COH is attached. Again, amine group is attached, hydrogen. And in the variable group, we can have hydrogen if it is glycine, or it is methyl, it is alanine, or it can be CH3COH if it is serine. That means we don't have any acid, that is acidic or basic group in the variable group, isn't it? So such amino acids where we don't have additional carboxylic acid in the amino acids. That means in one amino acid, if you consider, we have one carboxylic acid and one basic group. That is, amine is a basic. So please remember, amine is base here, and carboxylic group is a acid. If this is a condition and the R group don't have any carboxylic acid in it, then such amino acids, we consider it as neutral amino acid. Am I clear? What do you mean by neutral amino acids? An amino acid wherein we have only one COH and one amine group. See, the COH and one amine group is always there in any amino acid. Apart from this, if we don't have any extra carboxylic groups or amine groups in the variable group, then such amino acids, we call it as neutral amino acids. Clear? So examples you can remember like glycine, alanine, serine, which we have learned the structures also. Apart from that, we have valine. This is very, very important. So we have valine, glycine, and alanine. These are the examples for neutral amino acids. Very important. Clear? Now, glycine and alanine easily you can remember because we know the structure. Glycine has got only H and the R group. Alanine has got CH3 in the R group. Valine, since we didn't learn the structure, so you have to remember that valine is the example of neutral amino acid. Now, if you talk about basic amino acids, so what is the structure here? To one carbon, say we have COH here, then amine group here, and H here. Okay. Now, in the variable group, that is in the R group, if you have one more amine group, let us say CH2, and here we have amine group like this. So in this one amino acid, we have two amine groups, isn't it? So this kind of amino acid will be called as basic amino acid because it is alkaline. It has got one more amino acid. Clear? And examples you have to learn lysine and arginine. Very, very important. Basic amino acid, lysine and arginine. Next, coming to acidic, the same explanation. In amino acid, if you have the additional carboxylic groups in the variable group, then such amino acids, we call it as acidic. Very easy to learn. Aspartic acid and glutamic acid. Clear? Examples will be easy. Aspartic acid, glutamic acid, if they give you the same examples. But if they change the name of this aspartic and glutamic acid, then comes your problem. So aspartate. Aspartic acid is otherwise called as aspartate. Clear? So this you have to remember. And glutamic acid is otherwise called as glutamate. Clear? So here examples of these amino acids are very, very important from exam point of view. Directly they can give you the questions. For example of neutral amino acid will be your glycine, alanine, serine. Apart from that, valine. Very important. Basic amino acids example, lysine and arginine. For this, I can give you a trick like LA. LA is Los Angeles have got basic lifestyle. All right, Los Angeles, which is in your US. All right. So in that, Los Angeles is one of the state wherein we see basic lifestyle. Usually they don't have basic lifestyle, but still, in order to remember this example, you have to learn like Los Angeles have got basic lifestyle. So LA here is lysine and arginine. And then comes your acidic amino acids, aspartate, glutamate. If they give you aspartic acid, glutamic acid, very good. It is good. You can easily identify. But if they do not give, they give aspartate and glutamate. So please try to remember those terms. So this is one of the classification of amino acids based on the type of R group. That means if it has got the carboxylic group or the basic amino group. Next type of amino acids, we have alcoholic amino acids. So just now we have learned the example of serine, isn't it? Serine has got hydroxyl group. So alcohol here is nothing but OH. So if we have OH in the R group, then such amino acids are called alcoholic amino acids. Example will be serine. Very important again, serine is given in your textbook, threonine is not given in the textbook. So you can remember serine here. Next, we have sulfur containing amino acid, cysteine, methionine, very, very important. You can remember methionine will be always the first amino acid of the protein chain. This is also important. Whenever a protein chain is formed, the first amino acid will be the methionine always. So cysteine, methionine, sulfur containing, alcohol containing will be serine and threonine. Then neutral amino acid will be your valine. Then glycine, alanine, and your serine. The neutral amino acids. Okay. Then if you talk about the basic amino acids, we have Los Angeles, lysine and arginine, and acidic amino acids. You can remember aspartic acid, glutamic acid. Aspartic acid also called as aspartate, glutamic acid also called as glutamate. Next type of amino acids we have is aromatic amino acid. What do you mean by aromatic here? Aromatic is where we get the smells. Here, aroma. If you are getting some kind of smell, a particular smell, then we call it as aromatic. So here it is nothing but aroma, the smell. And aromatic amino acids, we have three, again, very, very important: tyrosine, tryptophan, and phenylalanine. Clear? Next, amino acids can be again classified into two types based on whether they are synthesized in our body or not. See, I said we have 20 amino acids in our body. Isn't it? There are 20 number of amino acids in our body which are involved in synthesizing of all kinds of proteins, whatever we have. But all these 20 amino acids are not synthesized in our body. Only few of them will be synthesized and few we have to take from our diet. Clear? So the one which are synthesized in our body, obviously they are non-essential. Can we say like that? But we have to say like that, isn't it? So here, the classification will be slightly confusing if you go through the terms. So we have essential amino acids and non-essential amino acids. Now, you have to remember the meaning of these two terms. Essential amino acids are not synthesized by the body. Clear? The essential amino acids are not synthesized by the body and we have to take them along with our diet. Whereas the non-essential amino acids are synthesized by the body. So does that mean that these amino acids are not essential for our body? That doesn't mean that. Clear? Please don't get confused by going with the meaning of the terms. You simply remember essential amino acids are not synthesized by our body. We have to take them along with our diet. Whereas the non-essential amino acids, they are essential actually. Clear? Non-essential amino acids, they are synthesized by the body. So we need not, bother. Since they are synthesized in our body, we need not take them along with our food. That is the meaning of these two types of amino acids, essential and non-essential. Now coming to the examples. Now examples are not mentioned in your textbook, but still, if you remember one column, then easily you can remember the other column. See, we have 20 amino acids which are classified into essential and non-essential. 10 amino acids are essential and the remaining 10 are non-essential. So if you remember one list, the other list you can tell, isn't it? So here I'll give you the trick to remember one column, that is essential amino acids. So essential amino acids are not synthesized in our body. We have to take them from the food. Please remember this. Clear? And what are the examples for this? You can remember this trick. Hill, MTV, tab. Clear? Hill MTV tab. This will be your list of essential amino acids, which are 10 in number. So what are all these things? H for histidine, I for isoleucine, L for leucine, other L for lysine. Clear? And then comes your MTV, M for methionine, and T. Please don't get confused. T is not tyrosine. It is tryptophan. This you have to remember. Clear? Because with T, we have so many amino acids. We have threonine, tryptophan, tyrosine. We have so many. So you can remember like 2 T's. We have the total 3 T's in amino acids. 2 T's will be your essential. Okay, only 1 T will be non-essential, and that 1 T is tyrosine. You can remember like that. So we have tryptophan, then V for valine. The other T will be your threonine. Then comes your arginine and phenylalanine. So this will be the list for the essential amino acid, and this trick you can easily apply in this. And whatever options if they are giving to identify the non-essential, you can simply remember this essential amino acids, and whichever is not there, that will be your non-essential amino acids. Clear? So this trick you can use it. Apart from essential and non-essential, we have one more type of amino acid based on this only. We call it as semi-essential amino acids. Clear? So if you remember essential amino acids are what? They are not synthesized by the body. Non-essential amino acids are synthesized in the body, whereas semi-essential, only sometimes our body will synthesize and sometimes we have to take from the diet. So this is your semi-essential amino acids. Example for semi-essential amino acids here I have taken. One is histidine and the second one will be your arginine. So here you can write it, histidine and arginine. These are the 2 semi-essential amino acids. Clear? So this is all about the classification of amino acids based on whether they are synthesized in the body or not. So this is all about the classification. Clear? Once more, if you quickly go through with the classification. First classification is based on whether the R group has got the carboxylic group or the amine group, additional carboxylic or amine group. So based on that, we can classify them as the neutral amino acids, acidic and basic. Clear? Examples you have to remember. Then we have alcoholic amino acids. Next, we'll have aromatic amino acids. Then we have all those things. And then we have, based on whether they are synthesized in the body or not, essential and non-essential. Apart from that, we have semi-essential amino acids also. Clear? Next, we'll talk about the properties of amino acids. Now, what is the property of amino acids? In neutral medium, I have written like it is acting as a zwitterion. So before that, let me explain this. What do you mean by all these things? Let me take a container. Clear? And in this container, I have simply introduced water. So water is a neutral medium, isn't it? So in this neutral medium, I have put an amino acid. So let me draw the amino acid here. COOH, NH2, H, and the R group. Now simply I'll write the R group because it can be any amino acid. Clear? Now in the neutral medium, what happens? From the carboxylic group, the OH will, the H will come out. All right. So we have H plus here. And from amine group, to the amine group, this H plus will go and bind. So what is the thing we get ultimately? Again, if I'm drawing a neutral medium here, the same thing. Okay, so here I'm going to show what exactly happens to the amino acid in a neutral medium. And this is the amino acid I have. NH, H, and R group. So here what happened? The H of CO, that is carboxylic acid group, one H have moved towards the amine group. So here we are left with COO, clear, minus, all right. And since instead of 2H here, we'll have 3H at amine group. So it will become plus, all right. So this side we have the anion, and this side we have the cation. That means a single molecule is showing two charges at the same time. One is negative and the other is positive. So if this is the condition of amino acid, such structure of amino acid, we call it as Zwitter ion. Am I clear? So what do you mean by Zwitter ion here? It has got both positive and negative charges in a single amino acid. And when do we get this Zwitter ion? When the amino acid is kept under neutral medium. This is the explanation for the Zwitter ion. Please remember, Zwitter ion is a molecule of amino acid wherein a single amino acid will show both positive and negative charges. And how do we get this? In neutral medium, the H of carboxylic acid will move towards the amine group. So amine group becomes positive and carboxylic group becomes negative. This is called Zwitter ion. Apart from this Zwitter ion, if you put now here in Zwitter ion formation, what did we do? We have kept the amino acid in neutral medium, isn't it? What happens when we keep the amino acid in acidic medium or in basic medium? That we'll see. So if you put the amino acid in acidic medium, let us say I am keeping this in acidic medium. So what is the acidic medium here? COOH, isn't it? And here in COOH, I'll have more and more of H plus ions. Isn't it? In acidic medium, we have more and more of H plus ions all over. And in this, I am keeping my amino acid. Let me write down this. NH2, H and R. So like this, now this is my acidic medium, wherein I have more and more H plus ions. Now what happens to this amino acid? This amino acid will accept the H plus ions in it. So it will get converted into, again, say this is my acidic medium. The COOH, NH, I'll write NH later. Okay. So whatever H plus ions were there, here before we have H2, is it NH2? Now, in this acidic medium, it will accept one more proton to become NH3. So when it is NH3, what is the charge here? Plus, isn't it? It is NH3 here when it is kept in acidic medium. And now this molecule of amino acid, we call it as base. Now we have NH3 plus. So this will be acting as a base. So this point you have to remember. When amino acid is kept under acidic medium, this molecule of amino acid will accept one proton to form a base. So acidic medium, it will act as a base. Similarly, if you talk about the amino acid when kept under basic medium, when it is kept under basic medium, it will act as an acid. Clear? Now here the explanation I won't give, or okay, I'll give what is there. So let me consider this one, COOH, NH2, then we have H and R. Clear? So when you are keeping in basic medium, what do we have? We have more and more of NH2. Clear? So here, the OH, the H of the amino acid will be taken by NH2. Clear? The H of COOH will be taken by NH2. As a result, we get COO minus NH2 will remain as it is, H and R. So here, this molecule will be acid. Clear? It is acid. So what are the points to remember here? When you keep amino acid in acidic medium, it will act as a base. Whereas when you keep the amino acid in basic medium, it will act as an acid. So it is acting opposite according to your medium. Clear? And such molecules which are acting oppositely, we call them as amphoteric molecules. So we say amino acids are amphoteric in nature. Okay, so these are the two terms you have to remember. One is amphoteric, the other is Zwitter ion. Amphoteric, you will see when the amino acids are kept in some medium, whether it is acid or basic medium. Clear? Zwitter ion is seen when the amino acid is kept under neutral medium. Difference you have to learn. Amphoteric, the amino acid will have only one charge. It can be positive charge or negative charge in a single molecule, whereas zwitterion is a condition wherein in a single amino acid will have both positive and negative charges. Such molecule, we call it as zwitterion. Okay, so this is all about the properties of amino acid. So till now, we have discussed the structure of amino acid, which is nothing but the substituted methanes by taking certain examples like glycine, alanine, and serine. Okay, then we have learned the peptide bond formation. Then we have learned the classification of amino acids, different types of amino acids, acidic, basic, neutral, alcoholic, aromatic. Clear? All these things we have discussed. And then essential amino acids, non-essential amino acids. And then we have discussed the properties of amino acid, wherein we have discussed the zwitterion and amphoteric nature of amino acid. Clear? Now, we will learn about the structure of proteins. So this was all about your monomer. Amino acids, we have learned that they are the monomer of protein. Now we'll learn what exactly the proteins are. So first and foremost, proteins are also called as polypeptides. What do you mean by polypeptides? Poly means what? Many. Peptide. Clear. Why do we call it as peptide here? Because in proteins always will have the peptide bond formation between the two amino acid. So since we have so many peptide bonds in a single protein molecule, hence the proteins are also called as polypeptides. Clear. Now we have one more term called oligopeptide. Now what do you mean by this oligopeptide? If you go through with the term, oligo means what? Few. If you remember the carbohydrates, we have learned monosaccharides, oligosaccharides, and polysaccharides. Do you remember? So in oligosaccharides, what we have learned here, if the number of monosaccharide are 2 to 10 in number, then such carbohydrates, we call it as oligosaccharides. Clear? That means few in number. So here also, if the number of amino acids is few, now what is the count? Now we are not sure, like it can be around, or we can say less than 50. If the number of amino acids are less than 50, then such proteins we call it as oligopeptides. If a protein molecule is made up of more than 50 molecules of amino acids, then that will be called as polypeptides. Okay. And one more very, very important, proteins are always heteropolymer. This point I have already explained. That means a protein is not made up of, it is never made up of only a single type of amino acids. A protein is always made up of different types of amino acids. Clear? And if you remember, the first amino acid will form the N terminal and the last amino acid will form the C terminal. Clear? These points are very important. And the most abundant protein in the animal world, if you talk, it is collagen. Clear? And the most abundant protein in biosphere, if you talk, it is rubisco. What is rubisco here? Rubisco is helpful in carbon fixation. That means photosynthesis. Okay, so rubisco is involved in photosynthesis, which is present in plants. So plant protein is the most abundant protein in biosphere, but specifically if they ask you what is the abundant animal protein, then you have to go with the collagen. So here, please don't get confused. All right, these points we have already read before also. The most abundant carbohydrate, cellulose, most abundant protein, rubisco, most abundant protein in animal world will be collagen. Clear? So this is the introduction part of protein. Next, we'll talk about the structure of proteins. So just now till now we have read that proteins are made up of amino acids, isn't it? Now we are going to learn how these amino acids are actually forming the structure. Like how does the protein look like? Clear? So here, if you talk about the structure of proteins, we have four basic structures of protein. So let me write down those first. First, we call it as primary structure. Second, we have secondary. Third will be tertiary. And the fourth will be quaternary structure. So these are the four different types of structures of proteins we have. First, we'll talk about the primary structure. Now, if you talk about the primary structure, this structure will give you simply the sequence of amino acids. That means, say for example, I have written like this. Okay, so first one is methionine. Next one is serine. Next one is tyrosine. Next one is tryptophan. Then next one is your phenylalanine. All right. Then, so like this. And all of them are bonded by peptide bonds. Clear? So let me complete this. Histidine and threonine. Okay. And then cysteine. All right. So say this is a sequence of amino acid. Now here I can say first amino acid is methionine, second is serine, third is tyrosine, fourth is tryptophan, like that, isn't it? So I can name like which number, at what number, which amino acid is there. So basically the primary structure of protein will give you the sequence. It is giving the sequence of amino acids. This point is very, very important. So primary structure of any protein will give us the information about at what sequence amino acids are arranged. What is the first amino acid, second, third, fourth, fifth, like that. Clear? So this is your primary structure. Now this primary structure will form the secondary structure. Now in this example, what I have drawn, all the amino acids are I have drawn linearly, isn't it? But throughout this protein, for example, this protein is made up of hundreds of amino acids. We have hundreds of amino acids in this protein. Clear? Now, this entire protein need not be made up of a single line. All right. In between, we can have secondary structures. So, how is the secondary structure formed? Let us see that. Secondary structure is of two types. It can be alpha helix or beta pleated. So, for this, I'll give you an example. Let us consider that I have given you a pearl chain. Okay, pearls. So, each pearl there will be telling you that amino acid. So each pearl is your amino acid. And now, so this sequence of amino acid or the sequence of pearl will be your primary structure. Okay. Now what you have to do is you have to take this pearl chain and you have to rotate it like this. So you will form the helix like this of the pearl chain. So here we have pearls. Say these are the pearls we have, and you are rotating in the form of helix. So this will form your alpha helix structure of the protein. Okay. Or else what you have to do is you have to make it zigzag. So instead of rotating like helix, you have just made it like a zigzag. Clear? Now here again we have the pearls all arranged in this. So what did I say? I have given you a pearl chain. So this pearl chain, which was a linear, is the primary structure of protein. Clear? Now this pearl chain, you have rotated it. So when you are rotating it, you'll get some spiral-like structure, and that will form your alpha helix. Instead of rotating, you are making zigzags. Clear? And that will give you the beta pleated. So either you can rotate it to form alpha helix, or you can simply make pleats to form a beta pleated. Clear? Now, when you are rotating it to form this alpha helix, can we have the stable structure here? No, because it tends to again get to that linear structure, isn't it? So in order to keep that pearl chain in helical structure, what you have to do, you have to fix some more, attachments there. So in this case, what we have to do is, what is done here is all this alpha helix will be given certain structure. That means it will be supported by certain additional bonds. See, already we have peptide bonds here, isn't it? Peptide bonds are always present in a protein because they are attaching the amino acids. Apart from this, what else? We have to give some additional bonds to support it so that this helical structure will not again attain a linear structure. So what are those additional bonds we have? Those will be your hydrogen bonds. Okay, and we have the disulfide bonds. Apart from this, we can say van der Waals forces. So these are the additional bonds, we can say, which will give the support to the helix, to the pearl chain, what we have. Similarly, beta pleated. Clear? So here, this will be giving the structure. Let us say these are the bonds we have between them, which are giving the support. Am I clear? So what happened till now? We have primary structure, which is your linear pearl chain. Each pearl in that pearl chain will be determining your amino acid. Now, what did you do? You have rotated that pearl chain to form the secondary structure. Example is alpha helix, or you have taken that pearl chain and you have made pleats to form beta pleated. And please remember, this alpha helix will be always right hand folding. Okay, the folding will be always towards the right hand side. Clear? Now, and in order to give support to that secondary structure, we have to provide certain additional bonds there. Clear? That will be hydrogen bonds, disulfide bonds, and van der Waals forces. So you can see the diagrams what you have what is given in your textbook. This kind helix like this is your alpha helix, whereas this is your beta pleated. Here, and for this, examples are again very important. Alpha helix, the spiral one, example will be keratin. Okay, so keratin is a type of protein which we find in hair. In our hair, we have a keratin protein which has got the secondary structure, and we have fibrin or fibroin in the silk. Okay, so fibroin is a protein which we see in silk, has got beta pleated. So please remember, I can give you a trick for this. Alpha helix, I told examples will be your keratin, which is present in hair. So usually some people will have curly hair, isn't it? I don't have curly hair, but some people will have curly hair. So that curls, you can resemble it with the alpha helix, and you can remember like keratin is present in the hair. Clear? And if you talk about beta pleated, I said it is present in fibroin protein. And where is this protein present? In silk. So we, while draping the silk saris or silk dhotis, we make pleats, remember, if you do it. Okay. So we make pleats of silk sari and silk dhoti. So like that, you can remember in silk or the protein, whatever is present in silk will have a beta pleated structure. Okay. So like this, you can remember the examples of alpha helix and beta pleats. Now coming to the third type of structure here, we have tertiary. Now what happens here? Now same thing. I have given you the pearl chain, which was linear, which was having a primary structure. You have rotated it to form helix. So that will be alpha helix, or you have made the pleats. That will be beta pleated. Clear? So alpha helix or beta pleated, both of them will be example for your secondary structures, types of secondary structures. Now what you have to do is, this helix or the pleats, you have to, randomly concise it. Say this is your primary structure of protein. Okay. Then you have made it helix like this or pleats. Now what you have to do is, these things you have to just, randomly fold it to make it small in size. So this structure, what we get is called your tertiary structure. So this one will have 3D dimension, isn't it? You have taken a chain, you have made it helix, and that helix you have, folded it randomly. So that will have 3D dimension. So this will be just like your woolen ball, clear, with 3D view or 3D dimensional protein. So such structure of the protein, we call it as tertiary structure, clear. And this tertiary structure, now this is given in your textbook. This diagram is given for tertiary structure. Now, when you fold this protein to form that woolen ball-like structure, again, there are chances of getting to that helical or the zigzag structure, isn't it? So for that also, we have to have additional bonds. Now, what will be those additional bonds? The same thing, hydrogen or the disulfide bonds. Clear? Here also, we have hydrogen and disulfide bonds. Please remember, very, very important. Now, because of this tertiary structures, say for example, this is our tertiary structure of the protein, we can have the crevices. Say this is your crevice. Okay, crevices are the gaps what we can have. So these are the crevices which we get when you fold the protein. All right. And because of the formation of these crevices, this tertiary structure proteins can act as enzymes. Now, enzymes, if you know, now enzymes in detail we'll discuss in this chapter itself, but enzymes will have something called active sites. Okay, and these are the sites where the substrate molecule will go and bind. For example, let us consider this pink one is a substrate molecule. So this substrate molecule is able to go and fit into that crevice. It is possible only because of the tertiary structure of the protein present in the enzyme. Okay, so that crevice which is present in the enzymes, we call it as active site. So active site are nothing but your crevices in the tertiary structure proteins when it is in the form of enzyme where the substrate is going and binding. Clear? So example are enzymes, and apart from that, you can remember myoglobin. What is myoglobin? Myo is what? Muscle. If you remember, we have hemoglobin. Hemoglobin is a blood which we find in the blood vessels. Clear? Myoglobin is also a type of hemoglobin, but this is found in muscles. All right. So this is all about your tertiary structure protein. Next, we'll talk about the quaternary structure protein, the last structure. Now, what happens in quaternary structure is you have to take more than one tertiary structure proteins. For example, say this is one tertiary structure protein. This is another tertiary structure. This is one more, and this is one more. Clear? Now, in this example, I have taken 4 tertiary structure protein, and all these proteins I am bonding. Clear? So, when more than one tertiary structure proteins are coming together, that will form your quaternary structure. So, protein has got more than one subunit of tertiary structure, then that will form the quaternary structure. The example for this will be hemoglobin. Hemoglobin, if you consider, if you split this term, we have heme and globin. So here globin is your protein part. Okay. In hemoglobin molecule, we have heme and globin. So heme basically it is formed by iron and pyrrole rings. Pyrrole rings. All right. If you talk about globin, globin is the actual protein what we get in hemoglobin. And in this globin part, this protein what we are talking here is of 4 in number. So we have 4 tertiary structured protein like this. 2 will be same, the other 2 will be different. So here, these 2 are your alpha globin. And here, these 2 will be the beta globin chains. And all these 4 globin chains, the 2 alpha and 2 beta globin chains, will be in turn attached like this. All right. So this structure we find in hemoglobin, and hence hemoglobin will be the example for your quaternary structured protein. So with this, we have finished the 4 different structures of protein. So just we'll recap it again by using our example. Clear? So the pearl chain, the linear pearl chain, what I have given you will be your primary structure protein, wherein each pearl will tell you the position of the amino acids. Now the same pearl chain, what you have done is rotated to form helix, or you have made the pleats to form beta pleated. Okay, so alpha helix and beta pleated ones will be the secondary structured proteins. Now in order to avoid the rewinding, that means again it should not come to the linear structure. So for that you have given some additional bonds to it. Hydrogen bonds, disulfide bonds, van der Waals forces you have given, so that will be in helices. All right. And now this helices or the beta pleats, what you have done is you have simply folded them to form a ball-like structure. So that ball-like structure have got 3D dimension, and that is your tertiary protein. This tertiary proteins will have active sites. The crevices in that will form the active sites. So it is helpful for enzymes. Clear? Apart from enzymes, we have myoglobin also as the example for tertiary structure proteins. Now, coming to quaternary structure proteins, more than one tertiary structure proteins, if you combine together, that will form your quaternary structure proteins. Example is hemoglobin. Okay. So with this, we have finished the protein. Clear? Now, last one more point, very important. It has been asked in your PYQs. We have something called Ramachandran plot. Okay. Ramachandran is a scientist, and he has given the structure of the protein. So Ramachandran plot is used to confirm the structure of protein. So here they will confuse you. Ramachandran plot is given to confirm the structure of proteins, lipids, carbohydrates, nucleic acids. Like that, they'll give you in the option. So please remember, Ramachandran plot is something related to proteins only. Okay. So this is all about the proteins. Next, we'll talk about the lipids in the next video. Thank you.